Hello, and welcome back to What is a Biology? I'm your host, as always, Mr. Johnny Hopkins, and today we're going to be covering some more genetic vocab, but today we're going to be doing something very specific, transposons. Transposons are something that are very intriguing in biology, and although they're on the fringe of what you might not need to know, they're still very intriguing and a very useful way of understanding genetic structure. So, what is a transposon, first of all? Then we're going to talk about how they exist in bacteria, and then how they exist in eukaryotes and humans, because we are eukaryotes. So, what the heck is a transposon? It's a mobile DNA element. So, it moves in the DNA. What does that mean? That means this guy, this transposon, can exist in the DNA, and it'll just take itself out and just go to a different part in the DNA. These, I believe, were stated by Watson Crick as the parasitic DNA elements. They have no function. They are only good for self-preservation. So they serve no real function in genes or anything. They just kind of, they just stay alive, doing their own thing, by just kind of running around and messing around with the DNA. And so usually what happens is that these movements of a transposon from one part of DNA to the other part of DNA is actually a fairly rare occurrence in the germline. It's usually only a 1 in 8 chance of it actually passing on from, say, myself to my offspring. And because of this, you've only got a 1 in 8 chance. It's also rarely harmful, because if you have a 1 in 8 chance of doing that, and then you've got 98% of our DNA being a non-coding region, it's probably not going to really actually make much of a difference. Sorry about that. Just from me to my offspring. And so there are two different ways for a transposon to actually transpose in the DNA. There's the direct route by using DNA, so it just cuts itself out and goes in. And they have the retro one, which uses RNA, which, like retroviruses, it's RNA to DNA. And so it goes into RNA, and then it goes reverse transcriptase, and it sorts it right in there. So, first up, bacteria. We have something called an insertion sequence, which is pretty much just a transposon type in the bacteria. You have, so abbreviated as IS, it's about 1 to 2 kilobases. And so it's in the middle of a gene, and so insertion into the middle of the gene is quite rare. So like this, but this is a 1 in 10 to the 5 to 7 chance of actually killing the cell. So actually these insertion sequences can also insert into plasmids. So you've got this whole weird combination of different things. And so how this occurs is also done by a transposase. It's an enzyme that helps carry out the transposition of the transposon. We're just going to keep saying transpose. Just use every word that has transpose, translocate, anything with trans in it is going to be said 50 million times. Okay, so the transposase will associate with the transposon, and then what happens is that it excises the transposons, keeps associated with it, and so then what happens is that the DNA gets ligated, and so we have the original point where the transposon existed. Then we translocate the transposon with the transposase. Too much. And what happens is that there's a staggered cut somewhere further along the DNA, it could be upstream or downstream, and then so there's a staggered cut, so we get those little sticky ends, and the sticky ends attached with the transposon, and then we have ligation, and then bam, transposon moved from this position to that position, moved from here to here, all by the effect of the transposase associated with the transposon, and then translocating, and then have transposase further inserting the transposon. Got a lot of those words. So, other types of them exist are like activator and dissociation elements in these bacteria in different cells. This is so the activator element is actually an autonomous, and so it just kind of causes mutation, does the transposing, whereas the dissociation element, the DS, only is activated when the activator is there. And so these guys in combination have actually led to a lot of different evolutionary techniques, and one of the most notable ones is actually the corn, where if you actually have seen old corn, there's actually a lot of different kind of colors and variations. That's because of transposons, these activator dissociation elements. So further on, we're going to go into eukaryotes, good old humans. Because who cares about bacteria? We are narcissistic people, and we want to talk about eukaryotes, because that affects me personally. So we have P elements. So these guys are just transposons, and essentially just DNA cut and paste, and they increase in copy number during the S phase, during the mitotic cycle, and trying to reproduce themselves, trying to make themselves more well known in the cell community. So these guys are associated with long-term repeats, LTRs, and not to be misunderstood, long-term relationships, which our bodies have a very long-term relationship with these transposons. So these long-term repeats are account for about 8% of the human genome, and they are some of the most common transposons, which are also retrotransposons, which use RNA to insert. One quick note is essentially this is a DNA, a direct transposon, whereas if we have a retrotransposon, essentially this guy just gets copied by RNA polymerase, and then you just essentially have the same exact thing, except the transposon exists here and then exists here as well, because we have the RNA associate then does reverse transcriptase and then inserts it into the sequence at a random point where there's the sticky ends available. So long-term repeats look like this. So you've got a long-term repeat, you have a gene, then you got the long-term repeat. And so, apologies, with that retroviral life cycle, we essentially have the retroviral RNA genome, and so the downstream LTR directs the RNA polymerase for insertion, where the upstream actually cleaves the RNA, the primary transcript and the poly A tail from it, such that the mRNA exits without the LTR, and the LTR can go in and do its transposon funky fun business. So there's also a couple of different types of transposons in the human and eukaryotes. You got the long interspersed elements, and then the short interspersed elements. So your lines and your sines. Lines are about 6 kilobases long, whereas short ones are about 300 base pairs. So long and short makes some pretty easy sense. Even though this guy's the same name as sine, we know it's got nothing to do with SOHCAHTOA. So going on, you have with lines, you have actually 3 different types: L1, L2, and L3. And fun fact, in modern eukaryotes, humans, L1 is the only one that exists still. It's the contemporary version. L2 and L3 kind of just exited a long time ago in the evolutionary chain, which we can use different sequencing techniques, and we found this out, funny enough. So, lines actually account for about 21% of the human genome, human DNA, that good old guy. And so, the lines, they have no long-term relationship, they have no long-terminal repeat. And this long-terminal repeat, so essentially it just transposes using RNA, using our favorite retro technique. And so, in the lines, we have actually 2 ORFs. ORF means open reading frame. Essentially, that ORF is essentially just a single protein that is made by by a gene. So you got 2 ORFs. You got ORF1 and ORF2. And these open reading frame 1, it's about 1 kilobase, so 1,000 bases, and it encodes an RNA binding protein. And that RNA binding protein, binds to the line. And at ORF2, it actually is about 4 kilobases long, and it encodes this homolog to a reverse transcriptase. So you've got something that binds to the RNA, and you have something that, takes it back from RNA to DNA. So it can go and insert into the genome, like this. Good old transposases translocating with the transposases. So, essentially the transposase and the reverse transcriptase. Then you have sines, which account for about 13% of human DNA. And because it's only about 300 base pairs long, how is it able to, go and be a transposase and also, be a reverse transcriptase? Well, fun fact, it doesn't. It actually just uses the ones from line to do that for just the reverse transcription and integration. So sines depend on lines to perform their actions as a parasitic sort of DNA. And a specific type of sign, about 1.1 of the 1.6 million signs, is an ALU element, which has, and these guys have a very specific single recognition site enzyme ALUI that will actually help insert it in and recognize these sorts of things. So, as previously mentioned, we have a 1 in 8 chance for a germline for passing it on from me to my potential offspring. So 40% chance of this 1 in 8 chance being a line 1, and then 1 in 8 chance of it, then 60% of that 1 in 8 chance of passing it on for it to be a sign element. And so it's not very difficult to see how, with a lot of time and a lot of generations, going from, say, 4 billion years ago to now, how these parasitic DNA elements could very much just go and mess with the DNA just enough to where it doesn't kill the cell, where it changes things a little bit here and there, then further mutations happen, and that causes a bunch of different evolution along the way to see how, oh, this is how some new protein occurs, how this protein gets made, and then so on and so forth. And one specific type of this evolution is also exon shuffling, where we have a recombination of the ALU elements in the introns with 2 different genes, and then you move it around, and then you have this gene be the same gene originally, and then with moving it around, they actually get 2 different exons, so you get 2 different domains, and then you can have further divergent mutations from there that cause these genes to encode for different little things that might be kind of associated as like RNA binding proteins, but one of them instead, where they can be restriction ones, and they then now cut different sequences or stuff like that. So that is everything about transposon that I'll cover. So just real quick, transposon is a mobile DNA element, and there are ones that exist in bacteria and eukaryotes. And essentially the way that they work is that there's a transposon, and in a direct way, when you have DNA, transposase associates with a transposon, excises it, translocates, and then it goes and inserts it into a different part of the DNA, whereas the retroviral, it just copies this, takes it around to where it wants to go, reverse transcriptases it, and inserts it up in there. And so I got activator dissociation elements that exist. You got P elements in humans, and got the lines and the signs in humans, with the sign special ALU elements that are most notable. And these guys have contributed to the evolution of all species because of their ways. So that is genetic vocabs. Next time we'll talk a bit more about genetic structure. So that's all. Thank you very much for watching.